Alrighty. So my name is Caleb, and unfortunately, I wasn't able to present with my group because I was dealing with strep throat. But I've had the pleasure to do a voiceover right now and present this online. So those are my group mates down below that helped me make this presentation. And our topic was on the mechanism of interferon alpha and HIV treatment. And to the right there, we have a depiction of interferon alpha within the JAK-STAT signaling pathway, which I will get into a little bit later. So let's begin. So HIV is a virus that attacks the immune system, specifically CD4 plus T cells, which are crucial for fighting infections. And over time, if untreated, HIV can weaken the immune system to the point that the body can't defend itself against common infections or diseases, which allows it to be one of these major global health concerns that we have right now. And while highly active antiretroviral therapy has significantly decreased morbidity and mortality, it doesn't completely eliminate the virus. And one of the reasons is that HIV persists in the body in the form of latent reservoirs, particularly in resting CD4 plus T cells. And these reservoirs are a huge barrier to achieving a cure, although we have had the chance to develop some treatments, but the virus integrates into the host DNA and becomes transcriptionally silent, meaning it's not producing any new virus and it's essentially invisible to both the immune system and antiretroviral drugs, making it a silent spreader also, which can be a dangerous aspect of a virus. But this is where interferon alpha comes in, and it has a broad antiviral and immunomodulatory effect and has been studied for its ability to reduce viral replication, modulate immune responses, and inhibit the establishment of latency. So let's kind of get into interferon and how it actually works at a molecular level. So interferon alpha is produced when interferon alpha is produced and binds to its receptor on its own, it activates a signaling cascade that leads to the expression of interferon stimulated genes, or abbreviated as ISGs. And these ISGs then produce antiviral proteins that interfere with the virus at multiple stages. And this activation happens through the JAK-STAT pathway. And specifically, when interferon alpha binds to the interferon alpha receptor IFNAR, it activates JAK1 and TYK2, which are two tyrosine kinases, and these kinases phosphorylate STAT1 and STAT2, which then dimerize and bind to another molecule called IRFN to form ISGF3. ISGF3 then translocates to the nucleus and binds to interferon stimulated response elements, which is abbreviated as ISREs, in the DNA, triggering transcription of ISGs. This results in antiviral proteins inhibiting HIV replication at various stages, which includes inhibiting reverse transcription, blocking nuclear import, interfering with integration, and preventing virion assembly and release. So rather than just one step, interferon alpha stimulates a broad and multi-layered antiviral response, which I will kind of get into on this slide right here, which is kind of just an overview of all the different players and the aspects that go along and play a part in this process. So, as stated on the other slide, upon binding to its receptor, interferon alpha activates JAK1 and TYK2, which in turn phosphorylate STAT1 and STAT2. And these activated STAT proteins dimerize and translocate to the nucleus. And this ultimately drives the expression of a host ISG, including ApoBEC3G and MX1. And both of these have different function in playing a part in reducing the HIV infection. ApoBEC3G is a cytidine deaminase that induces hypermutation of the HIV genome, which severely impairs viral replication. And MX1 is a GTPase, which plays a role in preventing the assembly and release of HIV particles. And with both of those, it really limits the ability of HIV to replicate and spread. So here we kind of have a description of the modulation by interferon alpha, a continuation of that. So 2 to 5 strands of obligatory anodylate synthetase, abbreviated as OAS, and the protein kinase PKR are additionally ISGS activated by interferon alpha that inhibits HIV replication by promoting the degrade degradation of viral RNA and blocking viral protein translation. The OAS family proteins are activated by double-stranded RNA, often produced during infection, and OAS enzymes catalyze the production of 2,5 oligodenylate, and 2 to 5A molecules then activate RNase L and endor- endorbonuclease that degrades viral and cellular RNA, which ultimately restricts viral replication and induces it into an antiviral state. And PKR is activated in response to double-stranded RNA, which leads to its autophosphorylation and subsequent phosphorylation of eukaryotic initiation factor 2 alpha, abbreviated as EIF2A, and global inhibition of translation, including HIV mRNA translation, and this ultimately prevents the production of key viral proteins like TAT, REV, and GAG, disrupting the viral replication cycle. And on this page, we kind of get into dendritic cells and T cell activation. So, interferon alpha also affects other components of the immune system, especially dendritic cells, which are antigen presenting cells that prime T cells to recognize and respond pathogens. And when dendritic cells are exposed to interferon alpha, they become more efficient at presenting antigen. So one key effect is the production of interleukin 12, which is abbreviated, as IL-12, a cytokine that promotes differentiation of naive CD4 plus T cells into Th1 cells. And this Th1 polarization is important because Th1 cells produce cytokines like interferon Y, which activate macrophages and support cytotoxic T lymphocyte responses. And both are critical for fighting viral infections like HIV. So overall, interferon alpha enhances antigen presentation and cell-mediated immune responses, increasing the ability of the immune system to actually control and limit HIV. And on this slide, I kind of get into that a little more. So it also leads into an increased production of pro-inflammatory cytokines like IL-12 discussed earlier. And Th1 cells secrete other interferons in the same class as interferon alpha, further amplifying MHC expression and T cell activation. And in chronic HIV infection, the immune system is often in a state of chronic activation, which can lead to immune exhaustion and dysfunction. But interferon alpha can modulate this chronic immune activation by enhancing immune responses and reducing immune exhaustion. And interferon alpha activates ISGF3, which stimulates the promoter region of CD38 and its genes that correlate with that, which overall induces expression. And persistent CD38 expression in HIV is associated with T cell hyperactivation, immune exhaustion, and disease progression. And interferon alpha can induce the expression of immune checkpoints via interferon alpha IFNAR1 and JAK1 and STAT3 signaling pathways. Expression can occur through a variation and variety of mechanisms. Interferon alpha signaling can ultimately be binded through ISRE like, which is abbreviated for interferon response elements motifs in the PDC-D1 gene promoter, which ultimately increases its transcription or activation of RF, R, IRF9, excuse me, and STAT1 directly and promotes PD1 expression on T cells. Interferon alpha can also increase PD1 L1 expression on DCs and macrophages, further dampening the T cell response via the same pathway. And this persistent interferon alpha exposure maintains high PD1 levels on CD8 and CD4 plus T cells, leading to T cell exhaustion. And modulation of these immune checkpoints by interferon alpha may help restore immune function and improve the body's ability to control HIV and the infection that comes with it. But this needs to be finely regulated as excessive immune activation can lead to harmful inflammation and tissue damage. So let's kind of get into the viral latency of it. So this is arguably one of the biggest challenges that the HIV treatment has because HIV latency occurs when viral DNA integrates into the host genome but remains transcriptionally silent, meaning no new viruses are made and the infection or infected cell appears normal to the immune system, so it's not treated within the immune system. Interferon alpha can help prevent the establishment of this latent state. It does not do this easily, but it does this by increasing expression of antiviral restriction factors, which ultimately interfere with early stages of the HIV cycle. And going back to APOBE3 C3G, it causes G to A hypermutations in viral DNA, rendering it non-functionally. So going back to its nucleotide bases, SAM HD1 limits the ability of deoxynucleotide triphosphates, which HIV needs to reverse transcription. TRIM5 alpha disrupts the uncoding process, destabilizing the viral core and preventing successful reverse transcription. And MX2 blocks nuclear import of the pre-integration complex, stopping viral DNA from integrating into the host genome. And by targeting these stages, interferon alpha reduces the chance that HIV can successfully hide in latent reservoirs, which is critical for developing any potential any cure in the future, which would be really important, but would be a difficult task at the same time be- because reactivation of latent reservoirs can also lead to viral rebound where the virus rapidly replicates and overwhelms the immune system. So, a small fraction of HIV infected resting CD4 plus T cells spontaneously reactivate, and HIV RNA is transcribed and translated into viral proteins. These new virions are assembled and then released from reactivated cells, leading to the spread of HIV to unaffected CD4 plus T cells, increasing viral load. And if ART is not reinstated, HIV levels rise exponentially, leading to immune system damage and a lot other more symptoms that are dangerous to the body and the overall immune system itself. So timing and control of interferon alpha administration are critical. And I kind of got into this earlier, but overall, interferon alpha inhibits the establishment of HIV latency. And it does this by increasing the expression of antiviral restriction factors. And these are the different players that I got into, whether it's SAMHD1, TRIM5 alpha, MX2, or APOBEC3G. So they each play a different part, but ultimately they they play a part in reducing the integration of HIV DNA into the host genome. And they do this within the early stages of HIV invasion. So, interferon alpha may also prevent integration through chromatin modifications, such as upregulation of histone deacetylases, which is abbreviated as HDACs, which promote a closed chromatin structure and increase expression of heterochromatin proteins, which reduce accessible sites for HIV DNA insertion. And a study by Azzoni actually found that interferon alpha can decrease the likelihood of HIV integration during the early stages of the infection, going back to all those different players that I said earlier that try to limit its early access within the genome and within the cells. But it may offer a potential strategy to limit the formation of new latent reservoirs and reduce the overall viral burden. So, some challenge and future outlooks for this specific treatment are very specific, and they have very specific challenges that may need to be overcome in the future for it to consistently work on a reliable level, per se. So it does have the potential for adverse side effects, including flu-like symptoms, fatigue, and liver toxicity, which may limit the tolerability of interferon alpha in patients. There's a risk of viral rebound if latent reservoirs are reactivated without proper control or ART innervation. And there's overall variability in patient responses to interferon alpha therapy, just like any therapy or medicine that anyone would take. So, factors such as genetic differences, the timing of therapy, and the stage of infection contribute to the varying of efficiency of interferon alpha. Therefore, further research is needed to understand the mechanisms underlying these differences and to identify biomarkers that can predict treatment outcomes. But these further studies and its future direction going forward should focus on the optimization of interferon alpha use and combination therapies, particularly in strategies aimed at targeting viral reservoirs and reducing the risk of viral rebound. So, with that being said, let's kind of get into the conclusion. So, to summarize, interferon alpha plays multifaceted role in HIV treatment strategies. It activates ISGs via the JAK-STAT signaling pathway, interfering with HIV replication at multiple steps and stages. It enhances dendritic cell function and promotes Th1 polarization, strengthening the adaptive immune response, and it reduces the likelihood of latency by upregulating key restriction factors. Despite these benefits, challenges remain, especially related to immune overactivation and the complex effects on latency. But with further research and careful clinical design, interferon alpha could play an important role in future functional cure strategies for HIV. But careful management is required to balance its beneficial effects with potential risks such as viral rebound and immune activation. And that kind of ends it. Right there are our references. But overall, I just want to kind of touch on that. I think personally, interferon alpha is a good direction for HIV treatment. And I think with its various factors, different and affecting different stages along the way and preventing certain stages from even happening, I think it's a good step within our society to kind of take to prevent the overall spread and treatment of HIV. So, thank you for allowing me to present this PowerPoint and presentation. And I'm sorry I couldn't be there again, but I'm glad that I was able to present the whole presentation as a whole. Thank you.